So we're really excited to start this next session on immunotherapy. We're switching the order around just a little bit. Dr. Holbrook-Court cannot be with us today, but between me and George and Ron, we will for sure give you a great overview of immunotherapy, why it's important, why it's exciting, and why we're doing clinical trials. So I'll hand it over to Ron.
Okay. That works. Terrific. I noticed a couple of you had questions at the end of George's talk. So two things. Later, we will have a full hour for questions at the end of the day of all of the speakers who will be available to you. We'll also try to save some time at the end of this session for additional questions. There are index cards on your table. So if you have a question you'd really like to have answered at some point, especially in the latter session, just jot it down. We'll collect them during the breaks, and we'll field as many questions as we can. That's usually the best part of the day. That is all these incredible questions and the kinds of things that you can hear from our panelists. I just want to say thank you, and please join me in some applause for George. That was the most terrific overview I had seen in quite some time. Even I understood it, so really terrific. So let me talk to you a little bit first. So we're going to move to research and some clinical trial work that's very, very exciting right now. Later in the day, we'll bring the medical oncologists back, the surgeons back. We'll talk more about current treatments. And in between, we're going to be talking about nutrition and strategies for living with neuroendocrine cancers as well. So let me tell you a little bit about the Caring for Carcinoid Foundation first, so you get the context within which our incredible excitement about immunotherapy has really mushroomed over the past year. We are the only organization whose mission, we are a private nonprofit organization whose mission is solely dedicated to peer-reviewed research that is focused exclusively on neuroendocrine cancers. Our name, Caring for Carcinoid, is a bit of a holdover from our history. You will see, some of you are aware of this, you've been helping us with this, that there will be a name change because clearly our research has encompassed the full range of neuroendocrine cancers and tumors of all types. And so thank you again for all of you who have supported us over the years. That support has enabled us to fund over $10 million now in research. And while it is true that not every major cancer center has a major focus on neuroendocrine cancer, I'm proud to say that we actually fund research at 7 of the top 10 cancer centers here in the US, and you'll see some examples of that in a moment, as well as cancer centers across the globe, and you'll see a little bit of that coming up as well. All of that work is overseen by a board of directors composed entirely of patients, family members of patients, or individuals who have lost a father, a mother, a son, or a daughter in some cases to neuroendocrine cancer. And I say that because we are so urgently focused on the work to make sure not only that it's good science, but that we approach this with a sense of urgency that those family members and patients really impart to me as executive director and our entire staff, and I might say our board of scientific advisors. George Fisher is the co-chair of our Board of Scientific Advisors. It is an incredible group made up of people from all different disciplines, all brought together by this organization to focus on neuroendocrine cancers. And if you could only, I wish you could all watch the process when they are reviewing what's next, what should we be doing next. We bring all of the researchers, not only our own, but others from around the globe together once a year to share not only published results, but things they're working at that aren't even published yet, because you may know sometimes research doesn't advance as quickly as it should because people want to make sure they publish before somebody else gets credit for the research. And we make sure that certainly the people that we fund are committed to sharing that kind of information because we believe that's the only way to accelerate the process to better treatments and cures, and that's why we're here. We are committed, of course, also to supporting patients and families through information sessions, educational sessions like this, and working and partnering with some of those very wonderful centers I mentioned. And again, we are very, very excited about moving forward with our immunotherapy. So I'm going to get to advancing. Great. So this shows a little bit of, I said over $10 million over the 10 years or so, and you can see how that's been increasing really with the support of patients and families who are supporting our organization. 100% of the donations from individuals go to the research. It is the generosity of our board and some of our corporate sponsors that support our small operation, and we're very proud of that fact as well. One of the things that's really interesting in this chart, if you look at the different colors, which are a little hard to make out in the bars themselves, in the early stages, virtually everything we were doing, I hope this works and you can see it. You can't see that. I need George's big green pointer. Oh, there he is. This is easier and better. Terrific. If you look here in the beginning, almost all this is called discovery. So when this organization started out, as George said, we didn't even know what we were looking for. It was like looking for a needle in a haystack. Where can you find the mutations? Where could you find the markers? Where could you find the clinical or biologic evidence as to what begins to answer the questions that our patients and families have? What caused this disease? Why do I have it? Why does it grow slow? Why does it then grow fast? All these basic questions that needed scientifically based answers. And as we progressed, in some areas, we couldn't find good researchers, even though we were offering grants internationally even, and didn't fit the level of competence and sophistication that said to our scientific advisors, it's not worth investing in, we got to get better ideas. And that really started to take off and really reached a crescendo, there we go, in 2011, when we had that Johns Hopkins result that George referred to earlier, the first mutations found in that case in pancreatic neuroendocrine tumors. It opened up the door, not only for our researchers, by the way, but for researchers in other cancers. It's very interesting. Many of the breakthroughs in more common forms of cancer often come from rare diseases. And so DAX and ATRX, the mutations that were discovered by that Hopkins team, have now been looked at for brain cancers and others as well. So we learn from other forms of cancer, and they're learning from us as well. But most importantly, we were able to take those results and fund 4 centers that are just completing the projects, modeling those mutations in those mouse models that George mentioned earlier. And we're going to have now at Stanford and MD Anderson and places in the UK and all around the country, those kinds of models to do the kind of preclinical testing on tumors that have those kinds of mutations as those ramp up now. So that's sort of the basic building blocks of using the science, open up the door, create the preclinical models. What's really significant, if you look at the last 2 years, and the light blue is what we call translational. This is the work that really is directly connected to trying to improve clinical treatment. And the most exciting of this right now is our immunotherapy clinical trials, which we'll be talking about in a moment. So what's happened over the years, and some of you were here a year and a half ago when Elizabeth Gold-Hirsch-Yelling got up here and challenged everybody with her $1 million challenge grant. She said, I want to see all of these things continue, but I also want to see the door open to new and innovative ideas from around the globe on clinical front, and especially was interested in what we were just beginning to hear about, immunotherapy. And so in addition to the proposals we receive and the ones we solicit through our Board of Scientific Advisors, we make available every year grants to the entire cancer research community through what's called the American Association of Cancer Researchers. Dr. Nakakura, who you'll hear from later today on the panel, a surgeon up at UCSF, has just had, if you follow our newsletter, some breakthrough results in working with patients who've developed resistance to one of the few available drugs for treatment and overcoming that resistance, which he's discovered some very important and positive results in the preclinical phase, which we can now see, is there an opportunity to expand that and provide all of you with yet another possible treatment if something that you've been on might not continue to work or the side effects are just too overwhelming. So very proud, especially in my tenure as executive director, to see us really move to a portfolio that's got this terrific balance of really solid basic science, continuing the work of finding the mutations, opening those doors in the preclinical, but also moving to the translational very, very strongly. And if you look at, these are some of the current projects in that translational bucket, if you will. Three of them, of course, are our immunotherapy projects we're going to be talking about in just a minute here. One at the University of Pennsylvania here. For those of you who haven't seen the video of Dr. June, I guess it was on HBO the other night. Some of you see there was a piece on HBO about cures for cancer, and the last third of it was featuring Dr. June, University of Pennsylvania. So we'll be working with them to take a treatment that's been astoundingly successful for some leukemia patients and doing the basic science to modify the T cells, your immune cells, to see if we can create ones that will also attack neuroendocrine cancers. And if we're successful in that, then we go to a preclinical phase and eventually, hopefully, to a clinical trial there as well. You're going to hear a lot more from Pam about the clinical trial here at Stanford in a moment. And then we've also contracted, now this is a researcher from the United Kingdom, so we sent out proposals literally around the world asking for the best immunologists around the world to give us proposals as to learn more about how neuroendocrine tumors and cells behave in terms of working with or against the immune system that would really inform the kind of things you're going to hear a lot more from Pam about. The rest of this, I want to tell you, really reflects the range of projects and the range of top centers that are really engaged in our work. So for instance, Michael German is also up at UCSF. He's found in working with diabetes patients some drugs that he believes may actually have a very positive impact on neuroendocrine. That's another grant that we received and are now funding through that AACR connection. So a very powerful way of opening up the door. Herb Chen, these are both last year's AACR grants, is at the University of Wisconsin. Long-time, very respected clinical surgeon in terms of neuroendocrine, working with his colleagues to deliver a new drug through using nanotechnology. And Diane Reedy Lagunas, doing personalized medicine at the Memorial Sloan Kettering, and also working with a physician from Germany who found in a very small clinical trial a new and improved, if you will, approach to PRRT, which is, as you know, pretty standard treatment in Europe, and we're still hoping to bring to the US. And so that small clinical trial will be starting up shortly as well. And then two more labs, one here at Stanford and one at Dana-Farber in Boston, looking at that immune environment in terms of immunotherapy. So a little bit of a sense of the range of things that are currently being funded, and of course, the follow-up to much of the successful models that we've developed to continue to look at the basic building blocks. So let me end with, and then turn it over to George and Pam, what our strategy is on immunotherapy. We had started to hear some of the stories that I'm sure George will relate to about a year ago about what was happening in the cancer research world. We had no evidence specifically about neuroendocrine, but we were hearing the stories about breakthrough treatments and cures in melanoma and lung cancer and some of these other kinds of cancers. And our board and our board of scientific advisors both said, look, we got to learn about this. We've got to bring the best immunologic experts in to speak with us. If there's a way to bring this to our patients, it's our duty to do so. And I'm very proud to say that we have taken a very aggressive approach. To board members who've been with this board since its inception have said over and over again, this is the most exciting thing that we've ever done. And it's the most ambitious as well. So basically, we're looking at a multi-pronged effort, 2 major centers doing 2 large projects, and then several centers. So far, we funded 3, really helping that work progress in terms of the scientific basis. But quite frankly, that's just the beginning. If there are new and better ideas out there in immunotherapy that show as much or more promise, we're going to be looking at those as well, which is one of the reasons we're trying to raise so much money for this purpose. 2 major projects. As I mentioned, I talked a little bit about the UPenn one. You can see that video through HBO, I guess, and we can send it to you if you want to see that link. And you're going to hear about something that's going to be, I believe, in the next quarter, launched right here at Stanford that is very, very exciting that Pam will talk more about. And again, it's a very ambitious agenda. We are very, very pleased that the TripAdvisor Charitable Foundation has given us a $1 million grant and challenged us to raise $1 million from our community to launch these initial projects. I'm pleased to say we're about halfway towards that goal. We're going to get there with your help. That's what it really takes. So with that, let me turn it over to George to talk a little bit about immunotherapy, why we're so excited about it, and then Pam will talk about what's going to be happening here at Stanford.
Great, thanks. I'll take that. Thank you, Ron. So I'm a medical oncologist, and the cure word doesn't roll off my tongue easily, because I know how powerful that word is. I know what it means to each and every person who has a disease that we don't have a cure for or has a loved one with that. So I try not to use that word, though for the first time in my career, I think that we have the expectation that that's a possibility. I can't say that we've cured anyone yet, but the leukemic patients that you'll see on that video, if you download it, we'll make it available to everybody. There's a 5-year-old who's now 8 who would never have been 6 if it weren't for the University of Pennsylvania treatment. There are lung cancer patients with treatments that I'll tell you about who are out 5 years that would never have been out 5 years with no evidence of disease as a result of immunotherapy. And we never thought that lung cancer would be a treatment for immunotherapy. And I'm kind of embarrassed to be up here talking about immunotherapy because I was one of the naysayers. I thought it was just a bunch of BS for a number of years. And yes, it may work for some very unusual diseases that might be prone to the immune system, might be uniquely susceptible to the immune system anyway, but that's not true anymore. We're seeing it for some other diseases, and I hope to share some of that. I don't know, just to start, I don't know whether it will apply to anyone with neuroendocrine tumors yet. But one of the advantages of being with a board that's nimble and a foundation that can fund different things on the fly is that there are enough successes in other tumors that we decided, well, why wait 10 years for them to work out the system? Let's go in and put our foot in the door now. Let's see what works here. Let's learn more, and maybe we can be part of making it work for neuroendocrine tumors. And so that's where we are today. So it's also a little bit embarrassing for me to talk about immunology. I'm not an immunologist. Hollander, who's on your program, was unable to make it today, so I've kind of butchered his slides, and I'm sure I'll butcher his talk. This is immunotherapy in the Science magazine, which is sort of the Bible for laboratory scientists. This year marks a turning point in cancer as long sought-after efforts to unleash the immune system against tumors are paying off, even if the future remains a question mark. But the emphasis is that we're seeing responses in tumors that we never would have guessed would ever respond to the immune system, and the question is, how can we build on those successes? So when I took immunology in medical school, there were just T cells and B cells. B cells were responsible for making something called antibodies. Antibodies are molecules that the B cells make, and those antibodies go through the system and can bind to viruses and incapacitate viruses. And in fact, some of the vaccines that you get for measles and whatnot rely on your body making antibodies that then circulate, that then get rid of viruses when you get infected as soon as they see them. So antibodies are part of the immune system. Now, you'll hear antibodies in a different context because years ago, 2 decades ago, we learned how to harness the immune system of mice and have mice make antibodies to human proteins. And when we use those antibodies, we call them drugs. So we can actually use immune molecules that we make in mice, and then we put them in people as drugs because they go and they attack a specific protein. The protein that they attack is called an antigen. So antibodies bind to antigens, and they bind like a lock and key. And so it's very specific. It's beautiful biology that your immune system has to recognize anything that might be foreign and get rid of it. Okay? Now, that's B cells. I'm not going to talk about B cells. There's also T cells. T cells are the cells that we always thought, well, those are the cells that could theoretically kill a foreign cell. So viruses infect normal cells. That's why you get sick. And if you have an infected normal cell, the virus gets into the normal cell, makes a lot more virus, and then lyses that cell, and then you have a whole bunch of virus that go to adjacent cells. So it's in the body's best interest to learn how to kill cells that are infected with a virus so they're not able to propagate that virus. And so that's what we thought T cells did. T cells could kill virally infected cells. And they would recognize the virus or some foreign protein on the surface of the cell and say, you're abnormal, you're out of here. Okay? So T cells would go attack things that were foreign, get rid of them. And for decades, we would debate, well, are cancer cells foreign? They arose from normal cells, but are they different enough that T cells might attack them? The answer now is definitively yes. The question is, how can we make them attack them better? But it turns out that there are more complicated things. There are something called dendritic cells, whose job it is, and in fact, I'll tell you that when we knew this, T and B cells, there were lots of experiments where people took T cells out of the body and they tried to put them back in the body in order to kill cells, and they didn't do anything. We've been working with T cells for decades, and they just didn't do anything. We've been trying to make them smarter in the laboratory, and they didn't do anything. But Mother Nature's smarter than we are. Mother Nature has dendritic cells that take up antigens from dying cells or infected cells or damaged cells, and then they give them specifically to the T cells in a way that the T cells will then learn how to act against any cell that has that antigen. So I think of the dendritic cells as the professors of the immune system. Their job is to look around, ingest a bunch of abnormal things, and then show those abnormal things in a very specific way in a lymph node to T cells that are waiting there to be educated. They're in a classroom with T cells. They literally get turned on by these dendritic cells, and then when they find something that they can react against, they multiply, and they leave the lymph node, they get in the bloodstream, and they'll kill anything that has that marker, anything that has that antigen. So it's a powerful system, and the question is, number one, is it working in patients? And number two, how can we make it work better if it is working? Macrophages are cells that are just the garbage cells of the immune system. They actually go around and they pick up debris. And they pick up debris and they get rid of it. And they can ingest whole cells. They can actually just swallow up 6 cells that are dying and say, okay, you're not doing Mother Nature, you're not doing this person any good. We're going to get rid of you and sort of recycle the good proteins and DNA, but just get rid of the 6 cells. So that's what macrophages do. This is what we used to know. And so I'm going to just concentrate on the T cells. I can tell you that there are trials and treatments that target dendritic cells, a vaccine that was approved for prostate cancer, that target macrophages. CD47 is a drug that's gotten a lot of hype that's being developed here to try to stimulate macrophages to help kill cancers. There are B cells that UCSF has a wonderful grant, and we're about to start another one, triggering B cells to help us fight cancers in different ways. But I'm only going to focus on one part of that, the T cells. So what are T cells? Well, it turns out it's more complicated. T cells are not just cells that go out and kill foreign things. There's actually a subset of, we call those killer T cells because they kill things. But there's also a subset called suppressor T cells. And sometimes they're known as T regulatory cells or Tregs. They look under the microscope like they're the same, but actually suppressor T cells have different markers that we can now recognize in the laboratory. And it's the job of the suppressor T cell to turn off the activated T cell or to prevent activation of a T cell. Why would you do that? Well, it turns out that Mother Nature has a certain number of ways of stimulating an immune response, but about twice that many ways of turning down an immune response. So in terms of evolution, it's probably more important for our survival that we can turn down an overzealous immune response than turn one on. And so there are all these mechanisms to turn off an immune response. Guess what? Cancer cells are smart. They have those same genes. Remember from the OJ world, you know, if the genes in your fingernail, in your hair, in your heart, and your eyeball are all the same. It just depends on which genes are turned on, which genes are turned off. Cancer cells start off with the same genes because they arise from normal cells. A cancer cell, all it has to do is turn on a gene that can evade the immune system. Now that we're starting to understand what genes are turned on in order to evade the immune system, we're able to give targeted therapies to prevent that evasion. Okay, so these proteins, these ways of evading the immune system are called checkpoint proteins. And the checkpoint proteins are on some normal tissues because if you get a good immune response, you don't want to be attacking normal cells. So some normal tissues will have checkpoint proteins, which Irv Weissman, a famous immunologist here, describes as if this is a normal cell and you have this checkpoint protein there, it's also called the don't eat me signal. And so your immune system's going around trying to eat up anything that looks abnormal, and it comes up against this and says, okay, I'm not eating that, because it sees this don't eat me signal. That's what a checkpoint protein is. And guess what? Tumors can overexpress don't eat me signals. And even if a T cell recognizes a tumor as foreign and is capable of seeing, look, that's a bad guy, I want to get it, if that tumor cell has a don't eat me signal, and there are a bunch of don't eat me signals, then that T cell cannot work. Okay. So it turns out that, as I said, antibodies from B cells, we can make antibodies in the laboratory, and again, many of our drugs, Avastin or bevacizumab is an antibody, cetuximab is an antibody, lots of antibodies. The first antibody ever put in a patient was here at Stanford, and that patient is still alive now 30 years later with a lymphoma. It was an antibody against that person's lymphoma. So antibodies have been around for a long time now and are very common ways of making drugs. We can make antibodies that bind to the checkpoint inhibitors or bind to these checkpoint proteins and block them. Now, what Ron was referring to is something completely different. And so I'm not going to talk about this, but those same T cells, if you take them out of the body, and if you know precisely what's on the surface of the tumor cell, like you can with leukemia and some lymphomas, you can genetically engineer the T cells in the laboratory, literally by changing the genes in the T cells so that they go after one specific antigen on any cell that has that antigen. And they'll go after it and kill it. And they're very good killers. The problem is they kill anything with that antigen, tumor cells or normal cells. So you have to be pretty darn sure that if you're going to go after something, you've got to make sure that you're not going to damage normal tissue. And so the kids who are cured, well, I'd like to use that word, the kids who are still in remission a few years later after that trial in the University of Pennsylvania have no B cells because the same thing that was on the leukemia is on their B cells. They have no B cells. We don't know yet what that will mean in 20 or 30 years for a kid who has no B cells. But the kid's alive with no leukemia. And so it may not be that simple to kind of bring it up to neuroendocrine tumors, although, as I mentioned in my kind of background talk, somatostatin receptor type 2 is on 80% of neuroendocrine tumors. We're trying to use that. That's what University of Pennsylvania is targeting with their CAR T cells. That's the technology used for this genetically engineered cell. And we may be able to show that we can kill all the SSTR2 bearing cells. The problem is we do have SSTR2 bearing cells in our pituitary, and we have them in other parts of the body, and the question is, what will that do? We don't know. So this is a brave new world. It's not going to necessarily be, okay, we got this, and that's the cure. There's lots of experimentation to be done, lots of safety things to work out. But like I said, I'm not going to be talking about that either. I'm just going to be talking about checkpoint proteins, which are easy. Checkpoint proteins are antibodies you can make in the lab. You know, the pharmaceutical companies charge $40,000 a dose for them, but they're actually relatively simple to make compared with engineering a T cell and making it specific for an individual. These are the same cells I just told you about. Let's call this gray one a tumor cell. So this is a tumor cell. We want to kill that cell. Well, this macrophage could just ingest it if it weren't for Irv Weissman's don't eat me signal, which he thinks may allow macrophages to ingest tumor cells. We don't know that yet. That works in mice, but we're just testing in people right now. This dendritic cell can take samples of antigens from the tumor cell and stimulate the T cell. This is the T cell in the middle. The T cell's in the middle because we think it's the most important cell to kill cancer cells. And so the dendritic cell might be able to stimulate the T cell to then go kill the cancer cell, providing the cancer doesn't have a don't eat me signal that the T cell recognizes. And I'll just tell you that PD1 here and PDL1 are the yin and the yang from these cells. So the PDL1 on the tumor cell and the PD1 on the T cell, if those two bind, the one thing we know is that T cell doesn't do anything. But if you don't have PDL1, then maybe the T cell has a chance of doing something. And if you block PDL1, maybe the T cell has a chance of doing something. Okay, so this is the science behind what I'm going to get to in a second. Another part is, I told you that there were Treg cells, suppressor T cells. Their job is to put the brakes on the immune system. They say, whoa, you guys are getting too active. We don't want to end up with an autoimmune disease, because you can die from an autoimmune disease too. We want to just put the brakes on the immune system. And so regulatory T cells can block T cells and keep them from working. Okay? So that's sort of a simplistic version, and the way they communicate with one another are through these little molecules on the surface of the cell that bind to molecules on the surface of another cell, and that's how they talk to each other. Well, remember, the cancer cells can show any protein they want. They just have to turn that gene on or turn that gene off. And if it helps them survive, guess what? They turn it on. So if we're smart enough to know which ones they turned on and which ones we can block to make them look digestible by the immune system or tasty, then we can get an immune response. So I'm going to focus again on T cells, but I'll tell you that there are a number of proteins. Some of them when you hit them, you can they're inhibitory receptors. So these proteins here block the immune response. And presumably, if you give an antibody, these are little cartoons of what antibodies look like. If you give an antibody and block that, then that cannot block the immune response because you're blocking the inhibitor. You're taking the brakes off. These antibodies, or these markers, actually stimulate the T cell. So that's why I say there's a yin and yang to it, that some things are turning up the activating or putting the gas pedal on the engine, and others are putting the brakes. And once you understand which these are, you might be able to make some headway. So is immunotherapy ready for prime time? I wish I knew for sure in neuroendocrine tumors. It certainly is in other cancers. This is a very important paper that demonstrated for the first time, the second time, but a more important drug for melanoma. So these are all just melanoma patients, okay? And this is a study that was done for Anthony Rebus from UCLA, but it was multiple authors. This is a tumor, a melanoma tumor, before treatment, at day 90, 3 months later. You can barely see it. That big gray mass is down to almost nothing. This is a big spot in the liver. All that dark area there in the liver is abnormal. Now it's down to the size of a ping pong ball. These were tumor cells. These were immune cells in the tumor at the baseline. These dark brown spots are immune cells in the tumor after treatment. So these are immune cells that are getting in there and killing the tumor. This graph is how we like to look at these things. Anything above 0 means that the tumor grew during the treatment. Anything that's below 0 means that the tumor got smaller. And if you look at minus 100, it means that the tumor disappeared. So I'd say that 2/3 of these patients had about 30–40% shrinkage of their cancers. And for melanoma, that's just phenomenal. I treated melanoma when I was a fellow. There were no treatments for melanoma. Even our biggest, nastiest chemotherapy drugs didn't do a damn thing for melanoma. And here we have a drug that has relatively few side effects that can cause complete remissions and a substantial proportion of patients who have shrinkage of their disease. This is another way of looking at it, which is equally impressive. The number of weeks on treatment. Every one of these bars is a single patient. So these are all single patients. Every blue arrow to the right means that patient's still on trial at the time of this publication in 2013. And you can see that this is 32 weeks, this is 50 weeks, this is over a year. A lot of people are on over a year with continued response to a treatment. Okay? So that's what we're talking about, the potential of the immune system in some diseases. Now, what about other diseases? So in my other life, I take care of pancreatic adenocarcinoma. Here are some patients with pancreatic adenocarcinoma. Now, not nearly as impressive, only 16 patients and only less than a handful But anything that works for pancreatic adenocarcinoma would be stunningly successful. And so I'm sorry, this is gastroesophageal cancer. So we just had a meeting in San Francisco, and a group from Japan showed that 30% of patients who had gastroesophageal cancers respond to the same drug. That's phenomenal. For chemotherapy, we barely get 30%, and chemotherapy is much tougher. Non-small cell lung cancer, a substantial proportion shrink. This is squamous cell head and neck, a substantial proportion shrink. Not everybody, so this is not a panacea. It's not going to be the cure-all for everybody, but they're seeing enough responses that it's got everyone excited about pursuing that and trying to improve upon it. Now, why might some people respond? Well, one of the questions is, well, maybe they're responding because there are lots of mutations. Why would there be lots of mutations? Here's a chart of different types of cancer based on the average number of mutations found in each cancer. And the one with the most mutations is melanoma. And maybe they're the most sensitive to immunotherapy because they have a lot of abnormal things that the immune system can recognize as foreign because they have a lot of mutations. That's possible. This was a slide from 2013. It's already out of date. At 2013, we had hints that this treatment worked for melanoma, non-small cell lung cancer, bladder cancer, head and neck cancer, some ovarian cancers, certainly some kidney cancers. So moving down towards less mutations, look at this carcinoid down here. But since then, we found that stomach cancer, it works for stomach cancer. It works for certain lymphomas very well. We're testing it now on glioblastoma, brain tumors. And we think that we can make it work for some breast cancers, and we're hoping to make it work for some pancreas cancers. So we're moving down to lower mutations. And if you look here, this is carcinoid. Carcinoid, as I mentioned this morning, has relatively few mutations. And so the question is, does it have enough mutations that the immune system will recognize it? We don't know, but we intend to find out. So with a low number of mutations, is that going to limit the effectiveness? We will only know once we test it. Why might some tumors respond to these drugs that target PD-1 and PD-L1, the inhibitory drugs, meaning the inhibitory proteins, the checkpoint proteins? Well, maybe the response is best in those tumors that have a lot of PD-L1. And so if you look at the PD-L1 positive tumors, I'm sorry, this is a little bit fuzzy, their response rate for PD-L1 positive tumors is higher than for PD-L1 negative tumors at every step, 36 versus 0, 67 versus 19, 44 versus 17. Now, it's not 0 across all of them, so PD-L1 negative tumors might respond, but PD-L1 positive tumors seem to have a better chance of responding when you give one of these PD-1 or PD-L1 drugs. And here's a list of the PD-1 drugs. This drug was just approved for melanoma in December. Was that 2 months ago? That's how new this is. This drug was approved for melanoma a few months before. This drug is CureTech in Israel. I'm not sure it's going to go anywhere. This drug is being tested in clinical trials, has fast-track status for a number of tumors from the FDA. Bristol has another drug. This is a drug bought by AstraZeneca. This is a foot race. Pharmaceutical companies are in this race as fast as they can to get these drugs to market because they work. We have to determine as a neuroendocrine tumor community how can we apply them to the diseases that we treat. I'm going to skip a couple slides here because these just go into how we measure the benefit, but let's just look at this. This is a situation in which patients received, at the start, this is a CT scan. Each one of these little arrows points to a little nodule. These are all, again, about the size of a golf ball, and this melanoma patient, and so this poor guy had a lot of nodules. 3 months later, 12 weeks later, there are no nodules. This is one of the patients, this patient is one of these points on this curve. The same thing I showed you earlier, 0 means the tumor didn't change at all. If it goes above 0, it means the tumor grew despite the treatment. And if it goes below 0, it means the tumor shrunk. This is a combination. This is not just the PD-1 drug now. This is the PD-1 plus another inhibitor of another checkpoint inhibitor, another checkpoint protein called CTLA-4 or ipilimumab. So now we're starting to say, if one drug works and another drug works, what happens if we combine them? Well, if you combine them in this particular study, you got a majority of people had fairly marked and rapid shrinkage of their tumors. And what I think is most impressive is that if you look at the people who had ipilimumab, which is the which it binds to CTLA-4, one of those checkpoint proteins, and nivolumab, which is an antibody that binds to PD-1, another checkpoint inhibitor, and you give them both together, you no longer saw the preference for PD-L1 positivity. So it worked just as well in the PD-L1 negative tumors. Why is that important? Well, we've already looked for PD-L1 in neuroendocrine tumors. We haven't found it. So neuroendocrine tumors are not chock full of PD-L1 like some melanomas are. But if this is true for melanoma, that you don't need PD-L1, maybe it'll be true for neuroendocrine, where we don't see PD-L1. Okay? So that's sort of the science in between some discoveries and then the application, how we justify trials. So if I link the dots, I would say that PD-1 or PD-L1 drugs might work better with a drug that targets the CTLA-4, at least it does in melanoma, especially in tumors with low PD-L1 expression. Okay? It might help those tumors that don't have PD-L1 expression. Maybe it's turning on PD-L1. We don't know yet. But what I didn't show you is that when you do give a combination of drugs, and it's true with almost all of oncology, when you give 2 drugs, you get more toxicity than when you give 1. And so these people had a fair amount of toxicity. The toxicity can be serious. It can be diarrhea. It's not just diarrhea like you get from some drugs. This is diarrhea from an autoimmune disease where your immune system is attacking your colon. We call that colitis. And people can end up in the hospital, and yes, there have been people who've died of autoimmune colitis or pneumonitis. That's where your immune system attacks your lung. So this is still, we have to go into this very carefully, very slowly, but that is one of the downsides of this combination treatment. Now we're learning how to give the combination safely, but it's still not the easiest of treatments. So Holbrook, who couldn't be here today, Holbrook Court, it's decided in the laboratory, well, if ipilimumab works great by vein, maybe it will work if we just injected it right in the tumor. Because all we want to do is turn off those T regulatory cells. That's what ipilimumab does. It turns off the Tregs, the T suppressor cells. And so if we give it right in the tumor and then we combine it with PD-1 or PD-L1, would that work? And in mouse studies I won't bore you with, he showed that it does work in the mice, and that's led to a trial that Pam's going to talk about. So there are lots of different ways of attacking the tumors with the immune system. One is with these checkpoint modulators. It's kind of the easiest way just because the antibodies are already in the pharmacy or they're in preclinical, they're already being made. So we know how to get these and we can use them just like we'd use drugs. Vaccines are a little bit more complicated. We need to manipulate tumor cells. We have to sometimes use viral vectors to get the vaccines there. So this takes a little bit more difficult to scale up. And the cell-based therapies too, the CAR T cells at University of Pennsylvania, that's going to be hundreds of thousands of dollars per patient in order to do that. But it's worth doing. It's definitely worth doing, and someday we'll make it cheaper. Okay, so it's tough to make predictions, especially about the future. My favorite, Yogi Berra, and I'm going to leave that now to let you hear about a little bit from Pam, and then we'll take questions after Pam's done.
It's George's right here. Great, thanks, George. That was a great overview of immunology, just to get us started and thinking about how this works in other cancers and how we might start applying it to NETs. So I'm excited to share with you a little bit more about our clinical trial using immunotherapy in NETs. I'm going to go over a few things, just talking a little bit more about clinical trials, just to give us all some background about how we design and think about trials. We'll talk a little bit briefly about what we've learned from recent NET trials. We'll talk then about the Stanford immunotherapy trial, and then how can you learn more about trials in general. So this is from the clinicaltrials.gov website, which is a good website to go to learn a little bit more about definitions and trials in general. But they define clinical trials as research studies that explore whether a medical strategy or treatment or device is safe and effective for humans. So that's just really the basic definition. I won't go, George and I share slides. So he went through and defined a little bit for you what phase 1, phase 2, and phase 3 clinical trials are. I'll remind you that every FDA-approved drug we have now had to jump through all of those hoops. So we have clinical trials to thank for the current and existing treatments that we have. In terms of designing and interpreting clinical trials intentionally have very specific eligibility criteria. We often want to study a very homogeneous or similar patient population in order to be sure that we know these results can be repeated. And in terms of defining success, we define success in a number of ways for clinical trials, and that's really tailored to the trial itself. Do we want to see if tumors shrink? That's a response rate. Do we want to see if we can slow the growth? That's progression-free survival. And do we want to see if it prolongs life? That's overall survival. So their clinical trials, the support really comes from 3 different mechanisms. One are investigator-initiated clinical trials. So these are trials where a researcher like myself or George may go to a pharmaceutical company and say, hey, I have a great idea. I want you guys to support this trial through the use of your drugs, and can you give us some money and some support? Industry-initiated clinical trials are trials that are really initiated by a pharmaceutical company, but they actually then need academic centers and other sites to actually implement the clinical trial. So they may come to places like Stanford and UCSF and others in order to get patients and enroll patients on these studies. And then there are cooperative group clinical trials. These are federally funded NIH trials, of which actually there are many for neuroendocrine tumors. These are when academic centers come together. And for example, there are a few with one with pancreatic neuroendocrine tumors right now of temozolomide versus temozolomide and capecitabine. These are often studies that we couldn't do just at a single center or through a pharmaceutical company, and we need the federal government to kick in, and those are wonderful mechanisms to get some important studies done. So why are clinical trials important? So clinical trials are a key research tool for advancing medical knowledge and patient care. Clinical research is done to really look at whether a new approach is safe and is it effective, and which treatments or strategies work best for certain groups of patients. So really trying to tailor treatments to certain niches. So as George had said, we know that some of these immunotherapies work in melanoma and in lung cancer, but it doesn't necessarily mean that we can apply it to others, but that may be some rationale for why we should study it. So interestingly, though all of our currently FDA-approved drugs had to go through clinical trials, only 3% of US adults with cancer participate in clinical trials. This is actually in striking contrast to the pediatric cancer population. And I don't know if George knows these numbers. 90% of the pediatric patient population participate in clinical trials. So I think that's a really key take-home point, and thank you to those of you who have already participated in trials. So I'll speak briefly about what we've learned from recent NET trials, because I think not only can we do really important large clinical trials in neuroendocrine tumors, but we need to in order to advance the field. So I'll go over 4 key trials just to set the stage for talking about the immunotherapy trial. So in 2009, the PROMID study was the first study that showed that somatostatin analogs like octreotide have an anti-cancer effect in NETs. In this trial, patients with midgut tumors were randomly assigned to receive octreotide or placebo. This is an example of a survival curve, and you can see that in the yellow line is above the blue line, which indicates that patients who got octreotide had more prolongation or more stable disease than those who got the placebo. So this is that progression-free survival parameter that I'd mentioned earlier. So lanreotide, George mentioned, is a new somatostatin analog, very similar to octreotide. This study actually looked at not only midgut patients, but patients with pancreatic, hindgut, and unknown primary NETs. This was a larger study of over 200 patients, just published this last fall, and in December, lanreotide was FDA approved for GI NETs on the basis that it also prolonged survival compared to placebo. So again, that top line in red is lanreotide, and the blue line is placebo, and it prolonged survival. So a third agent, so mTOR inhibitors, we've heard a little bit about everolimus. The brand name is Afinitor. This was a study published in 2011 where 400 patients with pancreatic NETs were randomly assigned to get everolimus versus placebo. And this was also a progression-free survival endpoint. Sutent prolonged progression-free survival. So patients had more stability compared to those who had placebo. This then led to the FDA approval of Sutent in pancreatic NETs. You can see we've actually, the last 5 years have seen incredible advances. For neuroendocrine tumors, certainly more so than in some of our other more common cancers like colon cancer. Lastly, so that was Afinitor. I may have misspoken and said Sutent, but the Sutent study was another phase 3 study that took patients with pancreatic NETs, 170 of them, randomized them to receive Sutent or placebo. And again, in this, patients had prolonged progression-free survival, and this also led to the FDA approval of Sutent. So since 2011, we have 3 new FDA-approved drugs. We have Sutent, Afinitor, and lanreotide. So that then sets the stage. We have, George actually mentioned also a couple of other clinical trials that we're eagerly awaiting the results we should hear in June, looking at bevacizumab, which is an antibody that blocks blood vessel growth to tumors, one in carcinoid and one in PNET, and those results should be presented at our big oncology meeting in June. So the Stanford immunotherapy trial. So George did a great job of describing these checkpoint inhibitors, and we plan to use 2 of them in a small phase 1/2. So phase 1 being we're looking at safety, and phase 2 we're starting to look at efficacy. And we're using 2 agents, the anti-CTLA-4 or ipilimumab and anti-PD-L1, which just has a alphabet soup as a name. It's not officially approved yet, but both of those in combination. So just by way of background, we need cancer therapies with less toxicity and that are more specific in targeting tumor cells. We already know that antibody-based therapies can target specific protein receptors on the surface of the cells. George mentioned that in his talk already. The first generation of these antibody-based therapies are already in clinic. So for example, bevacizumab, so a little note, so anything that ends in an AB is an antibody-based therapy. So bevacizumab or Avastin blocks blood vessel growth. It blocks that vascular endothelial growth factor protein. And that's approved for colon, lung, glioblastoma, others. Trastuzumab is an antibody that blocks HER2. That's FDA approved for the treatment of breast and stomach cancer. So these antibody-based therapies have been around for at least a decade. New therapies that target immunologic checkpoints in the patient's immune system to reverse tumor-induced immunosuppression. These immunotherapies have been limited historically to these immune-sensitive tumors like melanoma. George showed you that picture with all the tumors listed on the kind of horizontal line and the tumors that were more mutationally active versus those that were more mutationally quiet. So we want to see, are the more mutationally quiet tumors, can we use immunotherapies? And then the other background piece, immune-related side effects are potentially serious. George mentioned that as well. We certainly want to be helping patients and not harming them when we're introducing these immune therapies. NETs actually have some rationale for using immunotherapies already, though they're not really traditionally considered immune-sensitive tumors. Interferon alpha is actually a medication that's been around a long time, has been used for treating carcinoid tumors, and it's actually maybe the original immunotherapy. It's a naturally occurring cytokine, and interferon has been studied in many neuroendocrine trials. Okay, so our main scientific questions for this trial are, can we design a trial that, number one, has the potential to shift an immune-insensitive tumor such as NET to an immune-sensitive tumor? And can we limit the immune toxicities? So this is the schema and the study design, and I'll go over this just a little bit. So this is involving 2 medications, so as I'd mentioned, ipilimumab and the anti-PD-L1. So So all treatments are given in cycles. The cycle for this particular trial is given every 3 weeks, and the ipilimumab is given on day 1 as an intratumoral injection, so an injection into a single tumor site, whether that be liver, lymph node, or other. And then on day 2, patients will receive the anti-PD-L1 treatment, and then they will have a break for 2.5 weeks. Cycle 2, and on all subsequent cycles, they will receive anti-PD-L1 on day 1. So that intratumoral injection of ipilimumab is really, as George said, just to kind of stimulate the immune system, bring in those Treg cells. So the design of this trial, it's single arm, meaning all patients get exactly the same treatment. Phase 1/2, as I'd mentioned, testing safety and efficacy. We plan a very small pilot study of 20 patients, and then we're enrolling them in groups of 5 to really allow us to test for safety and be thoughtful as to how we enroll subsequent patients. The primary endpoints, as I'd mentioned, to assess safety and efficacy. We also have some really important secondary endpoints, which are to assess the immune environment and assess the immune response. Can we develop biomarkers for assessing that immune response? So I'll just remind you, so what are these drugs? So anti-CTLA-4 or ipilimumab, this is a liver with a tumor spot there. The injection is into the tumor itself. We actually we plan on giving less than what was given in the intravenous or IV treatments for melanoma in hopes of limiting toxicity. And then the anti-PD-L1 will be given intravenously. And I'll remind you, this is a slide that George showed. So the anti-CTLA-4 and anti-PD-1 or PD-L1s are in part of this blocking the inhibiting effect. So can we remove those brakes and really help the immune system recognize the cancer as being foreign and help fight it? So eligibility criteria, as I mentioned, are really important in terms of defining who's going to be on the clinical trial. Because this is really a pilot project, we're intentionally trying to be fairly broad in our eligibility criteria. About 10 years ago, when we were developing NET trials, we were lumpers and not splitters. And as clinical trials have progressed over the last few years, we recognize that pancreatic NETs are different than lung NETs, that are different than some other sites, and so we're starting to have more disease or site-specific clinical trials. For this trial, however, we are lumping again, and patients with both pancreatic NETs and non-pancreatic NETs will be eligible. We will allow all tumor grades with a Ki-67 up to 55%. So George mentioned grade earlier. So the grade 1 and 2 tumors typically have a Ki-67 of less than 20%. These are the well-differentiated or well-behaving tumors. Poorly differentiated tumors typically have a Ki-67 of greater than 20%. But I think what we're starting to learn is that cutoff is sort of arbitrary. Some of our colleagues in Europe have found that perhaps the lower end of that poorly differentiated group may actually behave more like well-differentiated tumors, which is why we're including those in this. Excuse me. The ECOG performance status is a way that oncologists and docs who do clinical trials assess a patient's performance status, meaning, what do you do on a daily basis? Can you climb a flight of stairs? How much are you out of bed during the day? It's really a global assessment of a patient's functional status. And so we want relatively healthy people who are out of bed more than half the day and can really tolerate a treatment. Patients must have had at least one prior treatment and have disease growth on that treatment. So there may be many patients who don't quite need this treatment yet. We certainly wouldn't want to stop a treatment that's already working for you in favor of an unknown treatment. So that's something, and I'll talk a little bit about that on some frequently asked questions. So these correlative studies I'd mentioned, Dr. Court's going to be running this aspect of the trial. Can we identify sophisticated immune markers of response? We're very excited about this aspect of the trial. I think we need to try to learn as much as we can about the neuroendocrine environment, the immune environment for neuroendocrine tumors. So frequently asked questions. This exists on the Caring for Carcinoid website, but I thought I'd just specifically address it here because we're asked about it a lot. So when is this going to start? We anticipate late spring, early summer of this year. How do I enroll? So really first discuss this with your oncologist. Do you need a new treatment? And if so, we're happy to talk to you, but I think the timing and choosing if and when you need a new treatment is important to do that with your treating oncologist. George and I are happy to see you at Stanford if the timing is right for you. Are there certain treatments or characteristics that would make me ineligible? So as I'd mentioned, the inclusion and exclusion criteria are important. I put the key eligibility criteria on that slide, but the full inclusion-exclusion criteria will be available once the trial opens. And then how do I consult with an oncologist at the center where the clinical trial is being conducted? You're, as I'd said, welcome to come see us at Stanford through our new patient coordinators if you and your oncologist feel the timing is right. And then lastly, how can you learn about clinical trials? So the clinicaltrials.gov website lists all clinical trials being performed in the US and some ex-US sites. However, it's actually kind of hard to navigate, to be totally honest. The Caring for Carcinoid Foundation has also put together a search tool for looking for neuroendocrine-specific clinical trials on their website. Stanford likewise has a tool. We also have a new app that you can download that helps look at Stanford-specific clinical trials. We have some information out at a table. This is our current list of clinical trials that range from clinical trials that address pancreatic NETs. We have the PRRT study open. George mentioned the neuroendocrine registry or database, which if you're interested, my research staff are around. But really, this is a way to help contribute. It's not a treatment, but it's if many of you here have already given us your clinical information, there's a questionnaire to try to learn more about environmental and family risk factors that we ask you to fill out. We use a piece of tissue, tumor tissue that was previously collected, so we're not asking you to take a new biopsy, and then a few extra tubes of blood. So in this way, we're really trying to take advantage of learning more about why patients get NETs. I'm starting to look at some biologic characteristics from those tumor tissues. So we would welcome your participation if you're interested. UCSF similarly has a great search tool on their website for clinical trials and NET trials. And they also have a number of great clinical trials. And I'll let Emily Bergslund speak to some of those later this afternoon. So in terms of my take-home points, I think I want to impart to you that there's really been an exponential amount of research in NETs over the last few years. It's truly exciting for both patients and for researchers. There are numerous active and developing clinical trials, one of which is the immunotherapy trial that we hope to open very soon. And immunotherapy, I think, holds considerable promise, both in other cancers, and we hope that it will have some application to NETs. So I'll stop there. I think George and I can take a few questions. We want Greta, maybe can come in, we can give you one of our mics. Greta McCare will give the next talk on nutrition, and then we'll break for lunch after that. So thank you.